Hi everyone, welcome back to our channel. Before we started, be sure that you subscribe to our channel by clicking on the button, subscribe button, on the below of this video. Okay, so today we are going to cover genetics chapter, which is chapter 8, Mendelian genetics. So breeding experiments of the Gregor Mendel in mid 1800s. Laid the groundwork for the science of genetics today. Okay, he published only 2 papers in his lifetime, and until 1884. Significance of his papers published in 1866 on inheritance and peace basically went unnoticed next 34 years until 3 separate botanists who also were the find out is actually heredity in plants. So we have a number of vocabulary including genotypes, phenotypes. Phenotypes is combination of genotypes and environment. So genotype determines phenotype. Environment also determines phenotype. Homozygous, recessive, dominant, complete dominance, incomplete dominance, codominance. Okay, so Mendel's experiment basically show us actually basic dominance, like his breeding experiments with cross-fertilization with purple pea plants with white pea plants, and F1 generation showing all purple pea plants, which is basically shows that it's actually it was completely dominant trait. So Mendel did almost 28,000 growth with 28,000 plants, and did study a number of traits including seed form, seed color, flower position, flower color, pod form, pod color, stem length, and then he showed that 3-to-1 ratio in single dominant genes. Okay, so now we know that if there is a specific location for each gene in chromosomes, it's called locus. Okay, Mendel focused on monohybrid 1-gene problems and dihybrid 2-gene problems. There is something also called test cross, which is defined as breeding. If you have an unknown genotype, you can actually cross that with a homozygous recessive allele and look at the offspring. If you see half of 1-to-1 ratio, that tells us that unknown is big W, little w, or heterozygous or carrier. Okay, so his 2-gene problems, like you see here, seed shape and seed color, actually give us in F2 generation 16 possible offsprings, and then a ratio to 9:3:3:1 ratio. Today there are some situations that are non-Mendelian, including incomplete dominance, like in this plant, which is called snapdragons. We see actually an intermediate color, which is pink, and then ratio 1:2:1. We also see in blood types codominance. If one parent has A and other parent has B, we see a blood type AB, which is a codominant allele. Codominant situation. Okay, A also completely dominant to O, B also completely dominant to O, but A codominant to B. So epistasis is we must have 2 genes, and then 1 gene is influencing the other gene expression. So you see here in code color of the rats, mammals, also another example is you can see here ratio actually is look like a 2-gene ratio, but it's actually a little off, 9:3:4. This tells us an epistatic situation. Okay, there is also polygenic inheritance, which is also non-Mendelian. In the polygenic situations, like skin colors in human or ear length in the corn, you can see actually there are several genes involved. It's not 1 or 2, but more than 2 genes involved. And this follows a normal distribution. You see here like a bell curve. And lastly, the environmental effect, which is basically, if not genetic, like in hydrangea plants, environment actually determines the flower color. If the pH soil is low, its flower color changes to the more different color than if soil pH is higher. Um, and also we have linked genes in plants, which means they are on the same chromosome, like you see in this tomato, chromosome number 1. Every gene you see here, they are linked because they are on same chromosome and they don't follow Mendelian genetics. This is also confirmed in Thomas Morgan's experiments, 1933 Nobel. This concludes our genetics chapter. See you in next video.